Hello, everybody. Thank you for being here. I know that we have to rush a little bit, but so I'm going to give you the message right up front, okay? Museums, natural history collections can actually inform how climate change is impacting biological forms of life, right? Big time. And we can do these now better than we have ever been able to, okay? I'm going to ask you to raise your hands if you know that we have a great natural history collection here at Texas Tech. Have you ever been to this? Okay. I see that two of them are biologists, so that doesn't count. But the point is that we have a great collection here, and the idea that natural history collections are those cabinets where all the specimens are resting it's long gone, all right? It has changed relatively recently, in fact. And this is a slide that counts by big numbers the number of specimens that we have here in the U.S., the number of collections that we have in the U.S. And up until about 10 or 15 years, all these were isolated collections with their own databases, right? But as part of the National Science Foundation initiative in digitizing collections, these collections are now online. That includes collections that we have here at Texas Tech, okay? And the idea that of this digitization process was to make certain that a statement that was presented by Joseph Grinnell in 1910 basically says the importance of our museum specimens are going to be realized not now, not in a couple of years, but maybe in 100 years, okay? And that's exactly what's happening, because every single one of the specimens that comes into museums is associated with data. It's associated with data in terms of geographic location, date, and in some cases, especially starting from the 60s and on, with genetic information, tissues that are preserved in very different ways, right? So he said these, or he stated this statement when he was looking at when he was collected in California around the Yosemite Valley, and this is a picture of the same place several years later, right? And the idea is that we want to know what's going on with those populations in those places, populations of birds, mammals, butterflies, you name them, because we can compare to what we have already in museums, because they are vouchered. Every single one of the observations that Grinnell made and that are presented in not only in California but all throughout the country are vouchered. They have dates. They have locations, and they have ancillary material. That means parasites, that means genetic data in some cases, okay? So basically this idea of using museums as a way to see how things are changing revolutionize what museum science has done, okay? And that is that vision that is being tried to be brought up by the National Science Foundation. And I'm a member of these, here throughout the National Science Research Laboratory, we're members of this program ID Bio, which is basically making available all the data of all the museums that want to participate. And the network is big, right? We have about 13 thematic collections that are centered throughout, they're distributed throughout the country. There's a hub in Florida, and all of these data is being made available to natural history, to everybody, okay? So it's no longer, so the access to the data is no longer closed behind museum doors. It's now broadly available, and you can go out and search for any taxa of interest and to see where were they located before, where are now present, and so on, and make ask questions about how is it that climate change may actually or may have impacted these populations. This is from a paper in 2006 that looks at how is it that climate change is going to affect a species, okay? And there's basically 3 scales of change. In words, that means that species either move or adapt or die out, okay? That's what's going to happen with populations, and some of these are species specific. What's important, for example, if we want to know how things are moving, how climate change is affecting specific species, is to know, for example, the species distribution. Where were they found? At what point in time? Can we build a niche envelope? Can we use Maxent to build up these predicted distributions for these particular taxa based on estimates of how climate is going to change, right? We need to know a little bit about the life history traits. How many offspring do they have? When is it that they have these offsprings? We need to know whether or not they move and so on. And all of those data points or data needs, if you will, that are highlighted by an asterisk come exactly from museum specimens, okay? So my goal, again, is to show you that museum science has evolved in a way that it makes these data available and allows anybody, not only trained biologists or ecologists, whatever, to ask how climate change is impacting natural species, okay? This is a great source for research in undergraduate education. So I also participate of an NSF workshop called AIM Up, whose goal is to use big data in the forms of these databases to train the next generation of biologists and ecologists, okay? And getting them access to these kinds of data sets. I have a couple minutes. I'm just going to point out a couple of examples. This is data from Berkeley and a colleague of mine who sent it over. These are data from Grinnell's collections from 1904 to 1939. They're trapped in several places where they generated maps of the distribution of the places where they were setting traps, for example. All these individuals, all the data, all the records, all the maps, all the field notes are in museums, and we have some of them, some similar data sets here. Okay, between 2003 and 2007, several groups went to see these particular spots where people had traveled from 1904 to 1911, and what they realized is that definitely with climate change, species are moving, they're shifting their ranges. In some particular cases, they're becoming narrow. The ranges are becoming narrow because climate is pushing them towards the limit of the distribution. In some cases, there's some ability to adapt. What's important is that this is species specific. Some species respond in one way, some other species respond in a different way. I want to end with talking about a couple of things that we've done here in my lab in terms of using museum specimens and their genetic data in the presence absence of virus data, okay, to predict areas that may be interesting to sample from an epidemiological standpoint. And what I'm going to do is I'm going to talk briefly in the next minute or so about hantaviruses. Hantaviruses are viruses that infect rodents and under certain circumstances can pass the species barrier and make people sick. Okay? These are viruses that one acquires by being in contact with dust where rodents defecated and whatnot, or if they're in contact with specimens, if they get a bite or a cut, they can get infected with this virus by touching these fluids. But what we want to know is if several strains of hantaviruses have their own specific species of rodents as reservoirs. There's a almost a one-to-one relationship, and people have suggested that these species of rodents and viruses have co-evolved for a long time. Okay, And one of the things we want to do is we want to see if we can predict where some of these hotspots for hantavirus outbreaks may be present. And we went to museum records, okay, museum here at Texas Tech and elsewhere, where we looked at the distributions of species that we know are harbor hantaviruses. And based on these data, on this point locality data, we extracted information from BioClim to build up these predictive models of distribution, and generated maps with probabilities of hantavirus outbreaks in South America for both rodents and humans. And then generated the maps of probabilities based on the presence of the rodents and the presence not only of the rodents but the rodents that are infected, the probability that some of these sites, we predicted the distribution of sites that may be harboring rodents that are infected with hantaviruses. I'm going to bypass these. The point is that we identified particular places in South America where we do not know, at least there are no reports of infection present yet, but every single predictive map suggests that we might be able to get rodents at least that are infected with hantavirus there. We wrote a grant to go this summer. We're going to be there in this particular part of the world. We're going to be testing these models. Now, all of these models are only possible because we have data from museum specimens that are vouchers, that are georeferenced, and for which we have ancillary materials. Okay, this is a very busy slide, but the only reason it's there is because the message I want to convey is that natural history collections have evolved by leaps and bounds beyond what they were even 15 years ago. And now natural history collections can inform how climate change is affecting the world on a very rigorous manner. And it can be done not only by trained biologists, this can be done by anybody who's interested and have access to internet. Okay, thank you. That's all I had to say.